we've up until now been mostly talking about a single species, looking at population density and how intraspecific competition. Now we're going to really shift our focus to interactions between species and how that influences population growth rates of both of them. And so we're going to be kind of looking at a much more, it's going to be sort of a more realistic look at how population regulation occurs between species that are often enemies of one another. And we're going to be looking at interspecific competition, predation, parasitism, and all sorts of things. Okay. So this is understanding how populations are regulated is important in especially a lot of things that there's widespread invasion of non-native species in a lot of ecosystems throughout the world. One of the classic examples, of course, is prickly pear cactus, which is not native to Australia, but it got there and it spread really rapidly. And a lot of times when you see new species coming to new places, they often undergo really high population explosions. And what do you think might be some of the causes of that? Why do you think invasive species, not all of them do well, a lot of them die out and never persist, but some of them, if they do persist, why do you think they kind of go through really rapid exponential growth? Yeah, it could be they don't have a lot of competitors around. What's the other thing that might allow them to really explode? Yeah, they might also not have a lot of enemies around besides just other competitors, but they may have left behind a lot of their parasites that regulate their population size, a lot of the predators that limit their population size. So invasive species are of considerable concern, and people in Australia, given all the native communities that are unique to Australia, and when prickly pears started to come in, this is what they did. So people were concerned, you're in these eucalyptus forests and things like that, and you basically have these huge swaths of prickly pear cactus that basically are preventing any sort of things from germinating, seeds from coming in, and all sorts of things, displacing a lot of the shrubs and ground cover and those sorts of things, and causing a lot of effects on the native communities in Australia. So the Australians didn't like that, much like they didn't like the Australian rabbits, right, and used myxoma. So the approach they used is they introduced, to the prickly pear cactus, they introduced a cactus moth that's a native, it's a thing that eats prickly pear, okay? So they introduced an herbivore of prickly pear, and look what happened. That's the cactus moth. They introduced it. Look what it did to the prickly pear. It pretty quickly eliminated the prickly pear from them and that sort of thing. Okay. So this shows that this was an effective biological control of an invasive species, okay? And this is just showing what happened immediately after the prickly pear was introduced and widespread elimination of this non-native species. Do you think that necessarily is the outcome? Yeah. So again, what you can imagine here is prickly pears, right? It may be that they all kind of went away, and then what do you think happened to the cactus moth when all of its resources went away? It might have gone to extinction too, right? And the problem could be that the cactus moth, maybe it can survive in various ways, maybe underground, it has root systems, it has various sorts of systems it can serve, even though the things above ground died, they may be able to come back. So a lot of things we're interested in is how sustainable are these biological controls, and prickly pear may be able to come back, and so how do you sustain these interactions between these sort of enemies? And so we'll talk about that. Okay. So how do we classify the types of interactions between species? And we can basically classify them as classification of types of interactions between species. So these are interactions between species. So what we're going to focus on today are what we call consumer resource interactions. And that's where one species by being in this interaction, there's a positive effect on it. The other species has a very negative effect on it, okay? And so the classic examples of consumer resource interactions is the predator, right? By having a prey has a positive effect on the predator. How does it do for the prey? Kind of a negative effect, right? You have, prey death, okay? Herbivores eating plants, right? The herbivores have eating the resource. They often have a negative effect on the plant. And then parasite-host interactions are another example of these sorts of consumer resource interactions. So what we're going to focus on, probably the next lecture or so, is on these consumer resource interactions between predator and prey, herbivore and plants, and parasite-host interactions. And then what we're going to move in towards our what we call interspecific competition, where both species are competing for a limited resource, and species 1 and species 2 both have a negative effect on one another. And this is what we call interspecific competition. So we'll be focusing the next few lectures on these things and looking at population dynamics in these kinds of contexts. So, yes. That's a wasp. That's a parasitoid wasp laying its eggs in probably a caterpillar larvae. Okay, so this is just an example of a consumer resource interaction. This parasitoid wasp lays its eggs inside this larvae. The larvae, the eggs, the larvae grow up inside of this thing and eventually kill that host. So this is a pretty dramatic example of a very negative. This obviously offspring survives, but a very negative effect on that those host larvae. So as we can kind of look at these, we can classify these consumer resource interactions along kind of two factors. One is kind of the duration or intimacy of the association between the consumer and the resource, and then the probability of death of the resource organism. And all of these consumer resource interactions can fall into one of these four cells, okay? So when we think about the example in this cell right here is where the probability of death of the resource organism is low. These are grazers and browsers, right? In general, bison or cows or things like, do they really cause the death of their host plant? Usually not. They eat it. They move on to something. They get it down to a very low level. But most plants and things like that, when they're being grazed or browsed, don't necessarily die in most cases, okay? They get eaten, okay? What's the duration and intimacy of the association between a grazer and a particular host plant individual? It's fairly short, right? A cow or buffalo or anything like that goes and grazes for a very short period of time and then moves on to another plant, okay, individual. So grazers and browsers have sort of low probability of death and a kind of a short and casual interaction. Predators and seed predators, things that specialize on eating seeds, are predators. What's the probability of death of the resource organism? A predator always kills its prey, right? That's what we call predation. That intimacy is fairly short and casual. There's active predation, consumption, and then they move on. Along here, we can also describe, and this isn't all, we'll kind of say this, the probability of death of the resource organism is fairly low for parasites and sort of arthropod herbivores, things that eat plant material and all those sorts of things. And the probability of death of the resource system in general is kind of low, but they often live in a fairly close relationship with the host individual, and it can be fairly long. They live in those things for a fairly long period of time. Parasitoids, the things I just showed, are kind of just at the opposite. Those things kill that host, kind of like predators, and they often have at least during the larval stage, a long and intimate association with it. Okay, so we're going to kind of look at mostly we're going to be focusing on predators and seed predators and parasites and effects on kind of population growth rates and population regulation and those sorts of things. Okay, so obviously I think we've talked a little bit about this. What can plants do, if you look at examples of insects laying their eggs on host plants, the larvae eating those host plants? What are the responses? Well, there are lots of different defenses or structural adaptations to prevent herbivory. Obviously, one of them are these sort of structural defenses, such as spines on this desert plant, right? That probably deters things from eating it or getting access to it and laying its eggs on it. We often, of course, obviously have chemical defenses. And this is an example of a milkweed plant, right? That has really nasty secondary chemical compounds, but obviously monarch butterflies have overcome those defenses. Their larvae can eat it, but they're about the only thing that can eat it, okay? So plants use a lot of defenses against herbivory and sort of structural and chemical defenses. And I mean, we've talked already a little bit about this. You know, you can induce chemical defenses. And so this has been done countless times. You can induce chemical defenses. This was an example in cotton plants where here's the thing that eats the cotton plants. It's a little mite that eats it. And if you have a control group that has control cotton plants that have never been exposed to that mite, and then you have a series of exposed cotton plants that have experienced being eaten by these mites, what happens is when in this exposed group, what you can see, those cotton plants that have previously been exposed to that mite, how do they do when they're then put on another cotton plant, or on those particular cotton plants that have been exposed? It actually turns out that the number of mites that are produced is much, much lower, right, in those cotton plants that have been previously exposed to this mite. These are controls that have never experienced a mite, and they have much higher numbers of adult mites and egg production. The basic take-home message is pretty simple. What happened here is these plants in this exposed group have produced chemical secondary compounds that are negative, and it influences the ability of adult mites to survive and also their egg production. Okay, so those cotton plants that have been exposed have chemicals in them that are defenses against these mites. And this has been shown in a lot of different kinds of organisms. Okay, so this is chapter 15. We'll kind of go into sort of we're going to get into now what happens to the population dynamics in these specific kinds of consumer resource interactions. So does everybody know what this is? Yep. And what's this? So yeah, it's a snowshoe hare. Okay? So there was a long-term collection of data from individuals collecting lynx and hare populations through the Hudson Bay, and this is primarily in Canada over long periods of time. Okay? So obviously this is a predator-prey interaction. This thing is benefiting, this one is not benefiting, right? And obviously there was a lot of people were collecting, during this period, collecting a lot of these lynx and snowshoe hares, and there are actually really good estimates of the population densities of these things over long periods of time, and people have continued to study these. So what we're going to look at here is an example, kind of using snowshoe hares and lynx interactions as predator-prey interactions as an example. So the thing we're going to focus in on, what factors influence the size and stability of these populations? And we're going to look at a bunch of different predator-prey interactions, and we're going to try to address two questions. Okay? So this is kind of keep this in the back of your minds as we go through each one of these examples. The first question we're going to try to get is, do predators, for example, the lynx, reduce the size of snowshoe hare populations below the carrying capacity set by resources for the prey, right? So when we think about this, when you think about that interaction between the snowshoe hare and the lynx, right, there's another element involved in that, right? And that's what? What are the snowshoe hares eating, right? And what do you think snowshoe hares eat? What do they primarily do? They eat vegetation, right? So when we think about it, we can think about this as kind of, from the lowest level, you're looking at the availability of plant material, right? Then how do hares influence that? And then how do predators respond to those lynx populations? So kind of keep this in mind. You have to remember there are three levels in this dynamic in studying this interaction and these population sizes. We're going to focus primarily on the predator and prey, but remember the resources that are available to the prey might also influence it. The availability of forage, vegetation, all those sorts of things can influence it. Okay. So one of the things we can do is, do prey populations such as the snowshoe hare, do they oscillate or change over time independent of predation? So the question is, can snowshoe hares go to high population level sizes, then decline, then go back up, that has nothing to do with predation? What do you think? What do you think? Yes. What do you think? We kind of looked at one of those examples of that, right? So you can have cases where this herbivore, right, it's a prey, right, is eating those resources. It can deplenish those resources, and it can cause a population crash. So all these changes, what we call oscillations, and all oscillations is over time the population goes up, down, in sort of regular cycles. The question is, are these oscillations primarily caused by changes in resources needed by the prey, or are the predators driving it? So we'll keep those things in mind, and we're going to spend a fair amount of time trying to figure this one out. So here's the data from the Hudson Bay Trading Company, where all these hunters came and they collected all these data, the population sizes of these things over basically 1850 to 1925. Okay, a couple things to notice. One along here is, this is the measures of population density of hares along this axis. And here's the population density of lynxes along this axis. What you'll notice is there are a lot more hares, right, than there are lynxes, and we'll eventually come to that. But one of the things we want to notice, does it look like these populations reach a stable equilibrium and maintain their population sizes at some constant value over time? Nope. What do you see? You see a lot of oscillations, right, where you go peak abundance, takes a little while, and then they crash, then they increase, then they crash, then they increase, then they crash, okay? And so look at the snowshoe hare example. From 1850, they were at fairly low population density. They went through an explosion, then they declined, then they went up, then they declined, okay? Look at the lynx example. Do you see the same sort of pattern? Okay, yeah. The lynx pattern, kind of ignore that one. The lynx pattern starts at low density here, and then you see this increase, then it declines, then it increases, then it declines, then it increases, okay? So these cycles, these are what are called fairly stable oscillations, right? And the peaks of these are fairly predictable. It's usually on about a 10 to 12 year cycle between the peaks of the population sizes. And it's fairly consistent over this time period, where you're always going to these peak levels about every 9 to 12 years, okay? So the cycles of the lynx and hare populations are highly synchronized. What do you notice about sort of where the peak population sizes of snowshoes, hares are versus where the maximum population sizes of the predator are? It's a little messy, but yeah, in general, good. Especially here, right? What do you see? Let's start at this point. What you see early on is a huge explosion of snowshoe hare, right? Do the lynx immediately increase in size right on top of it? No, it's actually lagged. This is what's called a time lag, where the peak in the population density of the predator is at some time later, okay? And you can probably figure out why that is, right? Yeah. When you think about it, which one of these things reproduces a lot faster and has shorter generation times? The snowshoe hares. The lynxes have much longer from the time a female is fertilized, her eggs are fertilized, and when she produces offspring, it takes a longer time. So it actually takes a while before, but you do see that as snowshoe hare population size increases, the lynx eventually does go up. So it responds to that availability of the prey density, okay? And you can also see that what's happening as those lynx populations go up. What's happening to the snowshoe hare population? It's going down, okay? Why do you think that is? They're getting eaten more, okay? So as the population density of that lynx goes up, higher predation rates on the prey, right, driving that population size down, okay? Some of these are kind of right on top of one another. We'll ignore those, okay? But over here, you do see these kind of offsets, right? Okay? So then this population is driven down, right? The prey population is driven down to very low levels. Then what happens to the lynx? Same thing, it goes down. So this is considered this is what's called these sort of coupled oscillations and with these time lags. And we're gonna come back to understand how that occurs. But people are still studying this in Canada and documenting the same patterns and continuing to study these things, and we're getting a much better understanding of what this. But this pattern has been demonstrated a long time ago. Here's a study that was done that many predator-prey populations show regular cycles of increase and decrease, these oscillations, right? And all we're showing here are the hare populations, so we're not looking at the lynx population densities at all. But the idea we're trying to demonstrate here is the amplitude or how high the peaks go may vary across environments, okay? So this was a study where they just looked at hare population density. That's all we have. We don't have lynx density over here. We have hare densities on two different environments. One is, make sure I get this right. Do I have this right? Yeah. So here's the mainland sites in this kind of orange color with the open circles. This is on the mainland, and the ones in red with red circles are on an island population. So again, you see these oscillations. Population densities of the hare are changing on both the island and on the mainland sites. But where are you seeing more dramatic oscillations? Which environment? Yeah, on the mainland side. So it goes up really high and then it crashes, right? Goes up, and then it crashes, goes up, and then it really crashes. So the magnitude of these oscillations is much greater on the mainland site. And on the island site, they go up, they go down, but you don't see these really dramatic declines. And the basic idea here is that actually, so the peaks of the hare densities on the island and mainland tend to coincide with one another, but the hare densities did not drop as low on the island. And it turns out what happened is, guess where lynxes are a lot more common? On the mainland. Okay, so the lynx density, and it's not shown here, is much, much higher on the mainland. The density of lynxes is much, much lower on the islands, and that's probably why it's not oscillating as much. Okay. Okay. Now we've just been talking about the lynx, the predator and the prey, the lynx and the snowshoe hares. Now let's bring in sort of the lower levels a little bit. Okay, so here's another example of an example of biological control. So this is actually still fairly commonly used in California. I don't know about Mexico in strawberry for organic farming. So the cyclamen mite, it's not shown here, is a major pest of strawberries, okay? And so a lot of organic farmers tend to use predators to control the population sizes of these mites that eat strawberries. Okay, so here's the predator that eats these cyclamen mites, so that it's the predator-prey interaction, but we're also talking about the strawberries. Okay. So let's look at what happens. And this is not as clean as the snowshoe hare lynx example, where you have these real tightly coupled oscillations. Okay, so again, this is done over about a 12-month period, just looking at the population density of both the predator and the prey. So the predator is on the red line, the cyclamen mite is the prey and the pest of the strawberry that the farmers want to control. And here are the population sizes of the prey or the pest. Okay. How would you describe that? I'd say messy. But in general, what you have is, let's start right here. Here you have the prey, right? You introduce the predator, what happens to it? It starts to decline, right? And what you can see is would you say this biological control agent, this predator, does a really good job of controlling the population size of that prey, that pest? Yeah, it keeps it at pretty low levels, right? So what you can see is with this predator around and it drives it really quickly to low levels, this prey remains pretty low. It goes up a little bit, but for the most part, it's really remaining at fairly low levels. Okay? So in the presence of the predator, the prey population in general remained relatively low. Okay? So one of the things you might want to think about is, so what's surprising about this then? So here's the predator. Wouldn't you expect it to just decline when its resource goes away? So it turns out it still remains fairly high levels of that predator. The population of the predator still remains relatively high, even when the prey has been reduced to a really low level. Guess what they do? When these things get to really low levels, guess what this predator does? No, well, yeah. Well, if they were eating each other, what would happen to the population density? It'd go down. It turns out what they do is they actually disperse and fly away and move away, and they eat other prey. So they can switch to other prey. They're not exclusively just eating this particular cyclamen mite. So they can maintain fairly high densities because they have alternative prey that they can eat in these agricultural systems. So they have a good chance. So they're a strong diversity. They eat other prey when cyclamen goes to low abundance. But as they start to increase a little bit, they'll come back and switch on them, eat them, and drive them back down to low levels. Okay. So this again shows that this predator can really maintain these pests at very low density. And this is considered one of the most effective biological control agents that people are using. Okay. Now let's look at what happens when you remove the predator from the system. So what they did, you don't want to know what parathion is, but they applied an insecticide that was specific to the predator. So they applied a parathion that reduced the predatory mite populations and pretty much got rid of them, okay? Or kept them at really low densities. So this is an example of where you just have the prey, the pest, and no predator. What'd you get? You got oscillations, didn't you? So here's an example. There's no predator in this situation, right? This is where they had to reapply the parathion on a fairly regular basis because these things kept coming back, okay? So what they did, they introduced the parathion, killed the predator. What happened to the population density of the cyclamen mite, the one in green? It exploded. Then what happened? It declined. Here's the parathion. They had the cyclamen. These predators were coming back, so they had to reapply. But anyway, what you see, it's kind of weird, but there were really high oscillations here. It got dampened a little bit, but you saw this continued oscillations of this prey population, even in the absence of the predator. Okay? So what do you think caused that? Here you don't have this predator. The prey, that herbivore, that pest, is only interacting presumably with this host plant, the strawberry, right? What could drive, weather, that's possible. Think of something else. Something about the host plant. Yeah. Probably one of the things that's going on is you're having a change in the resource availability, right? So this thing is going to high density. So there are two things that could happen. Why is the population declining after high density? What could the strawberry plant be doing? Yeah, it could be a chemical defense. Okay, that's one possibility. Okay. Can you think of anything else? Maybe it's depleting, its resource too, right? Maybe it's overgrazing, overeating the strawberries, and it's replenishing its resource. So it could be a chemical defense by the plant, or it may just be overeating its resource, and then its resource is crashing, and it crashes with it too. Okay. So I think I'm gonna leave it. Let me see where I am. Yeah, you can. Herbivory has dramatic effects on plant production. Okay. So this is just to illustrate. Probably one of the things that's happening here in this particular case is that the explosion in that prey population, the thing that's eating the strawberry, probably is causing a decline in the availability of strawberry leaves and things it can feed upon, right, and causing that decline. Okay. So here what you can see, here's a case where you got some oscillations but not very many. Here you got oscillations even in the absence of a predator, okay? So one of the things we'll focus on next time is trying to understand how can you experimentally test, what's driving oscillations. So think about at the top what we said in the snowshoe hare lynx example. Who did we say was driving the oscillations? The predator, right? What's this one saying? What's driving the oscillation? The resources, okay? So what we're going to try to really address, and there are ways to do this experimentally, and they did this snowshoe hare lynx example, what really causes snowshoe hare lynx oscillations? Is the predator driving it, or is it the food that the hares are eating that is driving it? So you have to kind of think about this as a fairly complex, at the different levels, predator, prey, plant. And the prey is in the middle, right? And which one is influencing the most, causing the oscillations? So that's what we'll try to do next time, and then we'll introduce you to this scary equation. Okay. More differential equations. So again, we're going to show a graph that illustrates how you can get oscillations under these models. So we'll deal with that next time. Okay. Oh, just a reminder, if you came in late, the homework is on Moodle. I'll send out an email with the link to the thing. The homework for the demography is on Moodle, and that's due Tuesday. What did I say, 10 points, 15 points? 15, okay.